Hello, and welcome to my presentation. Today I will talk about recent developments in the search for an effective vaccine against malaria. First, I will give an overview of the contents of this presentation. In the introduction, I will outline the scientists that contributed to the paper and give more details about the work in general, followed by a bit of general information on the pathogen, its life cycle, and the model systems which allowed the researchers to study the infection. After that, I will ask what could be gained by an effective vaccine against malaria. Once we all know the what and why, I will show how the authors have attempted, step by step through a series of experiments, to validate the viability, efficacy and safety of their novel approach, and after that, of course, the results and their interpretation. The paper. It was published in 2018, a mere 2 years ago, by a group of researchers, 25 in total, coming from a range of localities in Portugal, the Netherlands, Belgium, Spain, and the USA. The paper is an article, which means it presents empirical data to support a hypothesis. In this case, the authors have performed multiple different experiments in an effort to create a new live vaccine against malaria, and have modified a Plasmodium species that is known to cause disease in mice to do so. More details on that coming up soon. The parasite in question is Plasmodium falciparum, which causes the most lethal and relevant form of malaria. Here we can see a false color scanning electron microscope image of infected erythrocytes containing merozoites. Plasmodium falciparum is an eukaryotic intracellular pathogen, a protozoa and apicomplexa, which may infect hepatocytes and erythrocytes of its intermediate host, the human, spreads through the blood, and is eventually taken up by the definite host, a mosquito of the genus Anopheles. Here we can see the three main life cycles of the parasite: sporozoite and merozoite in the liver, then merozoite or gametocyte in the blood cells, and finally oocysts and sporozoites in the mosquito, which begins the cycle anew. As with each rationally designed medicine, we must now ask, which part of the pathogen do we want to fight? In this case, we have more than one life cycle stage which we can target. My paper focuses on the sporozoites in particular, and aims to prevent the first human cell infection, right after the mosquito bite. Another question we might want to ask is, do we really have to develop a vaccine against this disease, or should we shift our attention to a greater evil first, to save more lives? In the case of malaria, the answer is yes, absolutely. This is a disease very much worth fighting. This image shows the relative specific causes of death, other than risk factors such as air pollution or diet, ordered by their contribution to the total annual number of deaths in 2017. Admittedly, we need to search a bit before we find malaria. But what happens if we only look at the top 5 diseases which are definitely caused by pathogens? And now if we focus on those that are predominantly caused by one pathogen? Here we can see that creating a good, reliable vaccine against malaria might eliminate one of the leading causes of death in the world that can actually be prevented by a vaccine against a single organism. And it's not just that. The effects of malaria extend far beyond the number of deaths. Although the absolute number of malaria cases has decreased a bit in recent years, it is estimated that there have been 228 million cases globally and 405,000 deaths in 2018, so even though only a very small percentage died, the number of deaths still exceeded the total population of our university city by a factor of 2 in just 1 year, and many more suffered. Also, the burden of disease is spread very unequally. Most malaria cases occur in Africa and other poor countries, and most fatalities occur among young children. In total, it's estimated that 40% of the global population is at risk of infection, a number which may increase further with ongoing climate change. As we can see, there certainly is a need for a good vaccine. However, the experiments in my article did not make use of the Plasmodium falciparum parasite to formulate a vaccine, but rather a very similar organism. They used Plasmodium burgdyi, which causes malaria in mice and is shown as yellow in this SEM image. Plasmodium burgdyi is transmitted through Anopheles as well, and can infect humans to some extent, but not cause disease. Specifically, Plasmodium burgdyi is capable of infecting human hepatocytes, but is unable to develop in human erythrocytes, turning into a cryptic unviable form instead. And since the symptoms of the disease only occur during the erythrocytic stage, an infection with burgdyi should be unnoticeable by a human host. The authors used this information in a very smart way. Plasmodium organisms express a highly conserved protein, the circumsporozoite protein, or CS, on their surface, but only during the sporozoite and early hepatic stage. So they genetically modified PB to express the CS protein of Plasmodium falciparum instead, using the gene insertion marker out protocol, which makes it much easier to select for correctly transfected Plasmodium in culture, which resulted in a genetically modified Plasmodium species, naturally attenuated to express CS protein in human liver, but proliferate no further. This strain they called PB-VAC. To confirm their hypothesis, they needed model systems next. Here, they mainly used 3 types of model: mouse, rabbit, and tissue culture, or, more specifically, liver-humanized FRG mice, which are immunodeficient mice onto which human liver cells have been grafted, blood-humanized NSG mice, New Zealand white rabbit, because they display a Plasmodium burgeri infectivity pattern similar to humans, and a range of tissue strains, among them fresh human liver cells, mouse and human hepatomus and immortalized hepatocytes. Now that I've outlined the background information, I will present the experiments from the article. First, the sequence of the experiments, and what they wanted to achieve. I have identified 8 blocks of experiments, with each asking and answering a specific research question. Since they want to create an attenuated live vaccine, it's important to check if the modified organism only infects the right kind of cells in the right host, and delivers the epitope effectively. First, they compared the infectivity of PB in mouse and human liver cell culture, because else the expression platform wouldn't be able to deliver its antigens to a human host. Second, they tested if PB could infect human red blood cells in blood-humanized mice. Third, they performed calculations on the entire genome of both Plasmodium burgeri and Plasmodium falciparum. Fourth, they genetically modified PB and then checked if the resulting PB-VAC organism was still viable. Fifth, they compared the wild-type and modified PB in their ability to infect hepatocytes of mice and humans in tissue culture and humanized mice. Sixth, they examined the infectivity of PB in live rabbits, on rabbit liver and red blood cells. Seventh, they assessed the immune response of rabbits against the PB-VAC strain. And eighth and finally, they measured the inhibition of Plasmodium falciparum infection after immunization with PB-VAC. I will now quickly go over each experiment and its results. To check the infectivity of PB in liver cells, they used both in vivo and in vitro model systems. Here's a histological section of humanized FRG mice, with developing sporozoites visible both in the mice and human tissue area. And here we can see the relative amount of infected liver cells, and the development over time. Gray means mouse, and red human, and the results for the two are very similar. From this we can gather that PB has similar tropism and development in human and mouse hepatocytes within a humanized mouse. These results were further substantiated in human and mouse cell cultures. Next, they looked at the infectivity of PB in human red blood cells in the blood-humanized NSG mouse. After infection, they analyzed the blood in flow cytometry. And these are the results. They used 2 dyes, Cyto16 and TER119, which marked the nucleus and mouse erythrocytes, respectively. This way they could differentiate between human and mouse and infected and non-infected blood cells, as well as separate the 2 species erythrocytes magnetically. After which they looked at them under the microscope. They did find parasites within human RBCs, but only rarely, and always with only 1 nucleus, which means they did not develop as usual. Quantitative analysis showed that almost none of the human blood cells, here in red, got infected, in contrary to up to 5.5% of the mouse cells. After that, they calculated the predicted CD8-positive T-cell epitopes of the proteomes of both Plasmodium falciparum and burgeri and compared them. They found that over 60% of the epitopes were shared, which suggests a decent chance for cross-protection. Next, they observed the fitness of PB-VAC by following the development in mosquitoes. Here, the modified parasite was indistinguishable from the wild type, unlike previous genetic experiments which replaced the CS entirely with that of Plasmodium falciparum. PB-VAC expresses and sheds both variants of CS successfully during hepatic development. Then they checked the hepatic infectivity in human primary hepatocyte culture as well as humanized FRG mice. They found that PB-VAC as well as the wild type behaved similarly in culture, and caused similar parasite load in FRG mice, but could not infect human blood cells with much success. As the next step, they performed the infection experiments on rabbits, which are a step closer to humans as a disease model for malaria. Rabbit hepatocytes could be infected and develop merozoites, both in vitro primary hepatocytes as well as the live animal, over time and according to the parasite dosage. And just like human blood cells, the rabbit blood cells remain pretty much parasite-free. Now that we have established infectivity, we can look at immune response. For this purpose, rabbits were immunized multiple times via mosquito bite as a form of natural vaccine injection, blood and spleen collected, and IgG titer measured, using ELI. And Plasmodium falciparum or Plasmodium burgdorferi protein fragments as antigen. Compared to the control with no Plasmodium, or PB wild type, much more antibody bound to Plasmodium falciparum CS fragments when the animals were infected with PB-VAC. And lastly, the important question: can the antibodies against the modified PB strain prevent infection with Plasmodium falciparum? Since rabbits can't be infected by this pathogen, they used liver-humanized FRG mice and tissue culture as model system, and added isolated IgG antibodies. This setup showed that even a very low IgG titer already decreased infectivity in tissue culture by 40%, and a more physiological titer in humanized mice reduced the infection load to very low levels, while the control or PB wild-type control had little effect. Or in other words, a passive immunization was very successful. In the left graphic, you can see that the addition of antibodies against PB-VAC cut the percentage of falciparum infected cells in tissue culture almost in half, even though they used an antibody titer of only 0.2 milligrams per milliliter. In the mice, shown by the right graphic, they used 10 milligrams per milliliter, or 50 times as much, which is considered a physiological concentration. And this concludes the line of experiments, as well as my presentation. I think the results have shown that a vaccine against malaria might indeed be possible, with techniques and platforms we already have today, and considering the need, that is a very good thing indeed. After checking up on the authors of the paper, I sadly could not find anything pointing towards clinical trials. However, there were at least 2 preclinical evaluations on the topic of malaria vaccines, one of them discussing the method shown in this paper. And these are my sources.